Hey guys, today I'm going to talk about DNA replication. So this takes place where DNA is. Remember, the nucleus has nuclear pores that are small enough for things to go in and out, but not DNA. DNA cannot leave the nucleus, which means everything needs to go in the nucleus to allow replication. DNA replication is the multiplication of the DNA information. And it works not exactly as you would think it does. So the main analogy I want you to keep in mind is it goes from one double-stranded molecule of DNA, right? It opens it up and add new material on either side. So if you consider this is the book, it doesn't just copy the book and make a new book. It tears everything in half, so you have half a book on either side, and then recreates the other half. So at the end of the day, you are left with two double-stranded molecules of DNA that have half old DNA and half new DNA that we'll talk about in a second how it comes about. So really keep in mind that you tore the book in half. The information is half new, half old. So how does that work? Another analogy to help you understand how things start, well, the double strand of DNA is really compact. The adenosine is linked to the thymine, the thymine and the cytosine to the guanine, and all of those are really tightly packed together, and you need a zipper, like you would use to open up your zipper on your coat. And the zipper, it's the helicase. The helicase is that huge protein, always a protein and enzyme, and that helicase just opens things up. So it just zips along. And remember, the helicase goes that way. It will go and open up, and so you'll have more and more of the DNA opened up. Why do we need to open things up? We need the DNA to be viable to build a new one. To keep things open, because DNA naturally wants to rebind, to keep things open, we have the binding proteins. Those binding proteins here are just holding on to the strand of DNA, and those binding proteins prevent the closing. And so they're going to move along as well as the helicase opens up, the binding protein move along. Why is the DNA not closing back up? Because as soon as it's open, it allows for special fragments of DNA that are called primers, and they will start binding right there. But since the DNA keeps opening up that way, that strand, the primer, will just move, like stay where it is basically, but more DNA will be open, so I will be building with primer right there for simplicity. So the primer is just a short strand of DNA, short little sequence of DNA, right? And that little sequence of DNA is matching a little sequence of DNA on the existing strand. That's where we will start the replication. So on this strand, which is called the leading strand, so that's a primer here first, and the top one is called the leading, and this one is called the lagging, and it'll make sense in a second. So the leading strand of the primer stays where it is, and there's more and more DNA made available by opening up a molecule of DNA there. So there will be always more nucleotide here. So if we focus on top here and we take our DNA polymerase protein starts where the primer is at, and then we'll start moving along, adding nucleotide one by one, matching the nucleotide on the DNA and binding them to the primer, effectively making the new DNA. And so that DNA polymerase here is working continuously. Why is that? Because as I said, it keeps opening it up. It keeps going that way, but the zipper keeps going down the molecule all the way to the end, which means that polymerase always has new nucleotide. It always has things available to do that. The key about the polymerase is it always works from 5' to 3'. And as we know now, the DNA is counter-parallel, anti-parallel. So if that's 3', that's 5' on the complementary strand. That's a newly built strand, and that hand is 3' back there. That's great because the DNA polymerase goes 5 to 3. On the other hand here, the DNA polymerase here on the newly built strand goes still 5 to 3 on the newly built. So if I put my primer right there, and you'll see why I do that, my primer is right there, the DNA polymerase goes from, since 5 is here, that means the top here will be 3, and that means that one is 5, that means it goes that way, right? And so far so good, you're just like, yeah, what's the big deal? I'm just keep adding nucleotide and I go that way. And I do that with my DNA polymerase, being protein, right? Well, it's good, but remember, the DNA, the helicase goes that way, which means it's opening it up that way, which means that if you go to the right over there, it's actually closed up. So those two strands, the black strands, they meet back up and are closed, which means that that DNA polymerase is going to hit a wall. At some point, it's going to have to stop replicating. And that's why it's called the lagging strand, because at the end of the day, that polymerization of newly formed DNA is going to have to stop. And that helicase opens up more DNA viable that way, and you put a new primer and you start again. And your polymerase will hit the primer from the previous fragment, and you will be left with hanging fragments that are continuously lagging behind. And as we know from the PowerPoint, those fragments are called Okazaki fragments. And so let me write it. Uh, the lagging strand is matched with Okazaki fragment. And those Okazaki fragments have to be put back together with ligase, another type of protein. I encourage you to look at the PowerPoint for that. This video is really to emphasize the fact that the helicase moves that way, which by definition, since DNA polymerase moves from 5 to 3 prime on the leading strand and on the lagging strand as well, for the leading strand, it can't just keep going because the zipper keeps going that way. There's more and more nucleotide to work with. Whereas this, where on the lagging strand of the DNA molecule, it keeps going that way, but my polymerase can only go from 5 to 3, so it needs to go to the right and reach the end of the replication fork here, because those two black strands, the DNA closes back there. Which means that you make one fragment, then you go back there, you make another fragment, and the fragments are attached together with a ligase. Really important to recognize the leading and the lagging strand differences and the presence of Okazaki fragments.